Okay, so let's start the tutorial for single-cell RNA-seq analysis. So this class we will cover how you're going to analyze the single-cell RNA sequencing data set using the publicly available the resources. So this tutorial, we are sort of part of the learning RNA-seq and also how we can find the genes specific to certain cell types or certain conditions. And also we are looking at some examples of how we delineate the cell types from your experiments. So basically what we're going to use the Google Colab, which is the platform for publicly everyone can access to the codes or the research and bit of computing powers as well. So what you have to do is go to Google Colab on your web browser. So basically, you can use the Google Chrome or Safari or Microsoft Edge, and just type to Google Colab. Then that will list you to get to the website. Then once you get it, then you open up this notebook, which I show you in here. And also I'm going to share this one with you through the notification. Then once you get there, then log into your Google account and then you can use these things for your analysis. So what you can do is you can basically click the connect on here. So, this is the setting for Korean language. So, my apologies to different language for international students. But anyway, all the buttons are in the same, position, so you can find easily. So what you have to do is you can basically click these things. That's probably the connect on the English version. And in the meantime, we set up the computing on your Google Colab. So that's basically connect to your Google Colab, then see how many resources you can use. So you can see the RAM or you can see the hard disk, the amount of you can use for storing data or something like that. Okay, so basically Google Colab contains sort of the tutorial and also the background idea. What is the single cell RNA-seq analysis and how we can use the Python codes to analyze the data. So, I'm basically introducing both conceptual background and also the sort of basic hands-on tutorial for single cell RNA-seq data set. Okay, so what we're going to learn today is we're going to go through the pre-processing. Oops, I don't really like it. So let's use the draw. Is this working? Pre-processing? No. That would probably work. Okay. So we're going to go through the pre-processing of the PBMC dataset from the 10x genomics dataset. Then we're going to analyze the data with the Callisto and Bus tools we can get from the Python package. Then we're going to go through the analysis of identifying the cell types and their marker genes, right? And this notebook is based on previous the tutorials by Sina and Little. Then I further modified with the RNA-seq and single cell workflow. So two things we're going to learn today. So first one is the concept of RNA-seq alignment and file format. Obviously, once you learn about the procedures, then the modality of data is quite important. So we're going to learn about the file format as well. Then we're going to learn about the concept and also hands-on tutorial on a single cell RNA-seq analysis. Okay, so let's see the workflow overview of single cell analysis. So single cell analysis contains many different, the ways. So first one is the processing with the Callisto bus tools. So what you're going to do is you're going to do alignment of the sequence reads. We're going to talk about this one later. But this time we very specifically use the term, the pseudo alignment, which is sort of characteristics of Callisto tools. We're going to do the pseudo-aligned row sequencing reads to reference transcriptome. Then we get some, the gene quantification out of that, right? So you basically quantify the transcript abundance at a single cell level, right? So we're going to measure how much genes or transcripts expressed in single cells. So this is basically done by Callisto, right? And basically the steps is making read count, which is the number of reads we get from the sequencing data. Then there will be for individual transcript, and that will containing the gene expression for each cells, right? Then after that, we're going to go through the filtering and quality control, and afterwards, the Callisto boosters will convert the information into the count matrix, right? Then once we get these count matrix data, we're going to import the data into the Python environment. As you see here, we are all processing the data with the Python process. Then we're going to use the Scampi, which is very common tool for single cell analysis. We use the Scampi to process the single cell data further, right? Okay. Then afterwards, what we're going to do is we're going to do dimensional reduction and clustering. That's obviously used for the single cell data set, as it is high dimensional information. Then after that, once you reduce the dimensions, then we're going to visualization. Then you're going to do differential express analysis to find the genes very specifically expressed in certain cell clusters or certain experimental conditions, right? And afterwards, you're going to do some biological interpretation and visualization, and you think about the further analysis. So this is sort of the very basics of what we're going to do in this tutorial, right? Okay, so first thing you have to do is you set up the your Python environment. So what we're going to do is first we set up the time. So we basically want to measure how long it will take, right? So you measure the start time, then at the end of the code, you measure the end time, then you can see the entire process, how long it would take, right? So, I mean, I ran this one before. It takes about like a 35 minutes total. But this time when I show you the demonstration, it's a bit of mixed of, running codes and also, explanation. So it will take more than that. So just bear with me. Okay, so after that, what you have to do is you have to install the Python package, right? So you're going to install some of the package you require for this tutorial. So what you can do is you can basically press the button on here. So this run button, then it will do the installation, right? So you see here what this comment is doing. They say pip, we call pip, and this one is install. Install basically meaning that you, construct some software on your computer. But this time, we're not going to install the software on your computer. This is install the software to cloud computing, right? Because this is you, right? And you are using some, maybe notebook, my amazing artistic drawing, right? So this is your notebook. So you type this one, you're not going to install the package on your notebook. This is rather you install the package somewhere in the cloud. This is not literally cloud. This is, let's say, this is the Google Cloud. So GCP, we say, is a Google Cloud platform. So this is the data center somewhere in United States or somewhere in the Europe. So you basically type this command through the Google Colab, then you will install the package on the computer on the Google Cloud. So it's not gonna harm your computer anyway, right? Okay, so how we can install? You type the pip and install. This is the way you install the Python package on the certain environment, right? And what you're going to install, you install the matplotlib, you install the scikit-learn, you install the NumPy, you install the SciPy. So all three packages you are installed, right? So what you install further, you install the Leiden package, you install the Louvain and Scampi package as well. And you can type the pip, or sometimes you can specifically install certain versions. So pip3, which is you specify you want to install the package on Python version 3, right? So sometimes some packages not default to Python version 3. So in that case, you have to specify that. Yeah. And then once you install the package, what we're going to install further is, okay, before install the next software, you can see that the entire process of this install the package, you see how much the time you spent, right? So it's total like a 26 seconds, right? Okay, then what we're going to install further, we install the KB Python. So why it's KB Python? Because this is the Callisto bus tool, which we're going to use for the RNA-seq alignment. And this is the Python version. So Callisto bus tools, it has another version, not in Python, it's on the Bash or Unix. But this time we can very specifically use for the Python version. So we install the Python version like this. So you can just click the pip install and KB Python as well. So once you install the Callisto for your Google Colab, then we're going to download the data we use for the single cell analysis. So which data we're going to use, we get the data from 10x website, and the data called PBMC 1K version 3 FASTA-Qs. So this is the data containing all the cells and gene expression, but stored in a FASTA-Q format. Then once you download the data, then that will lead you to uncompress the data from your PAT file. Then we're going to use this command tar xf, then the data you download before. So that will take some time because the FASTA-Q files, generally quite large, so it takes some time for downloading. Then once you download the data, then what you're going to do is you have to use the index file for your reference mapping, because the RNA-seq data needs the quantification using the transcriptome dataset. Then in that case, you have to use the reference genome data, which we call the FASTA file. And this FASTA file also has the index file. So these two things are very common types of files used in bioinformatics and genomics. This is basically that I say is stored the biological sequence like a reference genome or reference transcriptome. So FASTA file, this is a little bit explanation about FASTA file. So FASTA file is a simple and widely used format for storing the biological sequence, such as the nucleotides, DNA or RNA. Or sometimes you can store the protein sequence as well. That's really up to your questions. And it has the two compositions. So first one is the header lines, which is the identifier line. And second one is the sequence data. And header line is begin with the greater than symbol and it followed by the sequence identifier or description. And this line provides information about the sequence such as name, source, and relevant annotation. And the sequence data is basically containing, after the header line, and this is actually containing the actual biological sequence. And the sequence is composed of the representing the nucleotides. So as we know that the ATCG and AUCG for RNA data. And basically the protein sequence, it's obviously storing the amino acid. And the sequence data can be split across multiple lines for readability, but it should not contain any space or breaks, right? So this is the examples of the DNA sequence in FASTA format. So you can put the header on here, and you can put a little bit identifier describing what you store in here. And this is the DNA sequence you actually want to store for certain information. And FASTA files are commonly used for storing sequence, genes, protein, genomes, or biological molecules. So there are also sort of standard format for providing sequence data for many, different bioinformatics analysis and databases. And FASTA files often come with the index file. So index file is in the context of genomics and bioinformatics. This is typically means that, auxiliary file generate to, facilitate rapid data retrieval from the large data source, such as the reference genome or large sequence data set. So as you can see in this example, and this index file work as a lookup tables that help locate specific pieces of information in the main data file quickly. And the format and content of the index file can vary, depends on the specific application and data type. So as you can see for the list of different examples, like a BAM index, we call BAI, and TABIX index, this is mostly for the VCF file you learned before. And FASTA index is FAI. And if you look at the sequence read archive from the public database, then it will use the SRA index, right? So this is the examples we can see for the FASTA file, but this time we can specifically use for the transcriptome FASTA, right? So this is the FASTA file, and this is the table file for T2G, probably means that transcript to gene, because the transcriptome looks like a information summarizing the transcript sequence per gene, right? So you have to have some table information converting transcripts to gene information. So once you make a index file, you have to use the Callisto tool. So basically the same command used in here. So KB, then you type the ref, then -d, which is, -d, which is, you specify it's human. And -i, you specify it's an index file.idx. So you're going to store your index file into this format, right? And this is, again, it takes a bit of time because it will go through the large data files containing the transcript information, we say transcriptome. Then once it takes some indexing, that will appear the index file at the end of the this running. Then after you get some index file, we are actually doing the alignment using the Callisto tool. So we're going to go through because it will take some time. So in a Callisto tools, we specifically call the alignment is the pseudo alignment, because Callisto tools use the alignment method, but it is based on the transcript level. And it looks a little bit different ways of alignment for RNA-seq quantification. So what is the pseudo alignment? This is sort of the concept introduced to address the challenge in RNA-seq alignment. And it offers a novel approach that focuses on aligning reads directly to a reference transcriptome rather than genome. So as you can see that previously people do the alignments to reference genome, and it's takes some time because that is the reason why we are moving on to pseudo alignment. And here are some key components of pseudo alignment. So if you look at the Callisto and Salmon, so pseudo alignment is primarily implemented using specialized software like a Callisto and Salmon. And the tools generate pseudo alignment based quantification, and it's basically faster and more accurate way to estimate the transcript abundances. And transcriptome index, it will do, it's not doing sort of aligning reads to the reference genome. It's actually doing the pseudo alignment, basically building the index of transcriptome as you see here. Then this index containing information about the transcript and their sequence and their abundance, and reads are then mapped directly to the transcript index, right? So once you map this sequence read out of the FASTA-Q files, then you're going to do making estimation of the transcript abundance, right? So pseudo-alignment 2 use the statistical methods to estimate the abundance of transcripts in RNA-seq data. And this is achieved by determining the compatibility of reads with transcripts in the index, right? So basically you are considering possible variation due to alternative splicing because, previously when you do the alignment of RNA-seq data, you are doing alignments to the gene, but this case you are align the reads to the transcript, so you can consider all the transcript, all the isoforms at the transcript level, so you can basically taking alternative splicing. So this is sort of the huge benefit you can expect from the pseudo alignment. So if you look at several advantages you can think about in an RNA-seq data analysis if you're using the pseudo alignment. So first one is the speed. So pseudo alignment is significantly faster than traditional alignment method. So that will making you more suitable for analyzing the larger data sets efficiently. And transcript level abundance basically doing, we have to go through one more lines in here, right? Quickly fix the block. Okay, so transcript level abundance. So pseudo alignment, as I say, it will do the quantification at the transcript level. So you can have more detailed and accurate characterization of gene expression. You can go through each transcript. And after that, you're very robust to alternative splicing. So as I mentioned that, if you go through the gene level, you're not going to see any isoform expression, but this time you are looking at the transcript, so you can basically taking all the alternative spliced form of the gene, right? And pseudo alignment is very good for the memory usage, which means that you're going to use less memory. So the low memory. So if you use the traditional alignment like a star, if you use those tools for human genome RNA-seq data, that takes like a 40 gigabytes for your alignment, or more than 40. But this time pseudo alignment takes only a few gigabytes. So this case, you're using the system RAM, which is around 12 gigabytes. And this time you have way less memory usage than previous method. Okay, so it still takes some time. Let's look at how the cooking is going. So you can start it by downloading some human files. It's automatically from the tool. and after that, extracting the file. So basically, you can use this information so you don't have to download the FASTA file by yourself. You can basically get from the server, right? So this is one beauty of we use the Google Cloud in your system. Okay, so after you make a index, what we are going to do is you basically do the run Callisto and Buzz tools, right? So this case, you can actually do actual RNA-seq alignment for your FASTA-Q files. So what I want to see first, a little bit dragging, but here we can see whether the data is really stored in here. So you can just type the ls, which is the comments to, oh, sorry. Go away. So type ls. That will show you the files on here. So that's probably the folder from this decompress. So you can see that if you see this folder, then you will see that the files on here, right? So you see how many files it has. This 1, 2, 3, 4, 5, 6. 6 files, right? And looks really similar, but they actually have different combinations. So one is the L001, and here is L002. So here is all L001, 002, and it has the I1, R1, and R2. So there is a meaning, right? So I will explain later, but briefly, R1 is the first paired end read, and this is the second paired end read, right? So that's why it's a read 1 and this is read 2. And I1 is the index read, so it has the index barcode and the data. So this is the one set of the data set, and this is another set of data set. You basically get the two set of the data set. But we're going to go through what is the specific information about this FASTA-Q format. So let's look at here first. So FASTA-Q, so let's look at the FASTA-Q first. So compared to FASTA files, so FASTA-Q files that basically containing the sequence information from the next generation sequencing dataset. So as you can see there, PBMC 1K with 3 S1 L001 R1 and 001.FASTA-Q file looks really crazy, but this is sort of very conventional naming you used in NGS dataset, and each part has sort of the meaning of the data. So PBMC 1K version 3 that's probably the PBMC 1K is describing the project name or the dataset name, and V3 is probably the version of the sequencing technology you are making the data. And S1 is the representation of the sample identification. So this case, probably just S1. Let's say it's the first sample. And L001, this is the representing the lane number. So if you do the high throughput sequencing like an Illumina machine, then you're going to use the multiple lanes. And most of the case, you're not going to use all of the lanes. You occupy 1 or 2 lanes. So in that case, you can specify the lane number. So lane 001 indicate this data comes from the lane 1. And R1, as I mentioned, that this is the paired data. So R1 is represent the first reads, so first pair, right? And 001 is representing the file number. So if you have a large amount of data, sometimes FASTA-Q file may be split into multiple smaller files, right? So 001 is the first file in the series, or 002, 003, something like that. You have the bunch of the data set out of your sequencing research, right? And the last composition is .FASTA-Q.gz. This is the file extension, so indicating the sequence data in FASTA-Q format, and it compressed the gzip compression. And FASTA-Q is the standard format for storing both sequence data and quality scores, right? So this is sort of the components you can see from the FASTA-Q files. Okay, anyhow, after you get all of that information, so we can basically run Callisto and Bus tools, right? And this comment takes a bit of time. I can say this takes like 30 minutes something. So once we run this information, we can go through all other information afterwards, right? So you can basically type the information KB count, which is you're going to count the data. So you're going to do quantification, right? So KB is saying Callisto Bus tools abbreviation, right? And you specify the parameter, which is H5AD. What it needs, this is optional parameters. You specify output should be in the H5AD format. So this is the sort of very specific format we are using for single cell data set because single cell data is large. So we have to have very efficient file format storing the large single cell data. And dash I is the same one. So this is which index you're going to use. So previously, when you make an index, you specify sort of the output names, for your index. But this time, you can just specify the generated index in this system because you are using index for quantification and pseudo alignment. So basically, same meaning. And dash G, T2G.txt. So this is the table format mapping the transcripts to genes, right? And 10xv3, this is you're going to specify you are using the sequencing data from the 10x genomics version 3. So one thing you shouldn't be confusing is we have the two different platforms with describing the sequencing. So here is the sequencing, the 10x genomics. This is basically you generate the sequencing library, right? Then once you generate the library, then you're going to sequence this library using Illumina sequencing, right? So 10x genomics is basically generate the single cell library. Then Illumina sequencing is the one sequence this library into the FASTA-Q format, right? So two different things. Don't be confused. And dash O is the output. So you can just specify the folder directory called output. And dash filter is you're going to filter some of the lower quality automatically. You can specify these parameters, but you can also use your, manual way to filter the data set. This will be covered by tutorial. And dash T is the, you specify the number of thread or CPUs you're going to use for the parallel processing because most of the case, computer has more than one CPU core, so you can do the multi-processing, which makes you much faster data process than the single core, right? So how many cores we have in this Google Colab? We cannot see from here, but I know that this contains the 2 cores in this free version, right? So you can use that. And as you can see here, the pseudo alignment takes a bit of RAM, nothing like a 1 gigabytes. It takes like an 8 to 12 gigabytes. So that's surely due in Google Colab. So that's a good thing, right? So you can freely use this resource. So how you can do the sequence alignment. So you use this index information. So you get all this FASTA-Q information mapped into the reference transcriptome, right? So this is the way you get from that process. So it takes some time. So we move on to explaining the different parts. So once you run the pseudo alignment, you get some output, but before we talking about the outputs, you can do sort of use different methods for alignments, not just the pseudo alignments. So which alignment tools you can use for pre-processing the single cell data? We can think about the three different things. So first one is the cell ranger, and second one is the star solo, and the last one is the 11, right? So Cell Ranger is sort of the suite tools developed by 10x Genomics, right? So you remember the 10x Genomics, they making the single cell library, but also they provide you the softwares you can use for the data set, right? So this is really good, right? And this is the company developing the single cell technology. So once you make a library, it automatically process the data with their software, right? So Cell Ranger is specifically designed for processing data generated by 10x Genomics Chromium platform. And it includes the generation of libraries with unique molecular identifier. This is very important concept in single cell RNA-seq data. We call UMI. And this UMI will be on the each cell, right? And this cell ranger is commonly used for pre-processing the 10x genomics data and suitable for users who require user-friendly and streamlined analysis pipeline. So Cell Ranger, I mean, I really go into this detail about their software, but it's really easy to use. And once you run that, then you can see, all in one result within the HTML file. So, which is good. But tutorial has to be a bit lighter than, Cell Ranger. So we go with the Callisto tool. And Star Solo is part of Star Alignment toolsuite. So basically, the very single specific tools part of the Star Alignment tool, which is very named for RNA-seq analysis. So Star Solo is designed for pre-processing the single cell seq data and be used for both droplet-based and plate-based single-cell experiments. So this is compared to Cell Ranger. They generally use for any kinds of single-cell experiments, so which is very, generous method. And 11 is part of the Salmon project and designed for efficient and accurate quantification of transcript abundance in single-cell data. So 11 is not a competitor of Callisto, and because they actually doing the pseudo alignments as Callisto, right? And these pseudo alignments are raised to transcriptome instead of aligning to genome, making computationally efficient, right? So which tools you have to choose, it really depends on you, and it really depends on the data set. So there is no really gold standard, for certain tools to process the single cell data. Of course, each tool say they're the best in the field, but I can see that there's a bit of pros and cons, right? I like to use the Cell Ranger because it's really, generally used in many single cell contexts, and also the 10x genomics is really the big platform in the field. But I can see that the Callisto or 11 is really good enough to process all the, the large scale data set with very light away. So there are also the things you have to consider in your process. But I mean, this time we only go through the Callisto example. Okay, it still takes some time, so we're going to go through the next part. So once you finish the Callisto pseudo alignment, you get some, the output, right? So you can type the ls, then output directory. Then you see each output on here. And if you specifically see count underscore unfiltered, then you can see this directory. You can see the information about the single cell quantification. So we have the two different versions. So one is the filtered and the different is the unfiltered. So filtered one is, as I say, it automatically done by the BOSS tools developed by Callisto tool team. But for the tutorial, we're going to use very specific, thorough QC. So we start from the unfiltered data set, as you see here. Okay, so once you get the data, so you can have many different options to process the single-cell RNA-seq analysis, right? But two major tools very popular for the single-cell RNA-seq analysis. One is the SCMP, and the other one is the SERA. I mean, the space is Chirat, but this is actually the French artist, so we call them SERA, right? So SCMP and SERA are the popular single-cell tool. They are not really the Python package, so sorry for the misinformation here. So they're the two popular packages used for the single-cell RNA-seq analysis, and they provide a wide range of tool and functionality for the pre-processing, quality control, visualization, clustering, downstream analysis of single-cell data. And we have different pros and cons for the SCMP and SERA. So what is the SCMP? This is the open access Python package, specifically on a efficient analysis of large-scale single-cell data set. And it builds on the scientific computing libraries called NumPy and SciPy and scikit-learn. So these three Python computing libraries, they make a easier use of and integrating many different data sets in your single cell analysis, right? So SCMP is basically following this flow, and this flow is basically same as the SERA as well. So because they are meant to be doing the same thing in single cell. But what's the big differences between the SCMP and SERA? SERA is based on the R, so it's R-based toolkit for the single cell analysis. I would say probably the SERA is easier than SCMP in terms of usability, because I think most of the tools in R is easier for biologists because it's really, you can see sort of the visual way in an R studio, then you see how the things is going on in your R studio. So SERA or the R package is generally easier than Python package, I guess. But when I can say you're not going to use the very lighter or the smaller scale of the single cell data set in your career. So at some point, you have to switch to some, the softwares or the ecosystem can process the large scale data set, right? So in that case, Python is much better choice than R. So in this context, the SCMP is probably better for SERA. This is my perspective. And SCMP is also not really terrible for beginners. So you can easily start your single cell analysis or sort of the programming skills using this SCMP. So that's why I choose this method for the tutorial. So the Callisto tools is still going on, so we can just go through the next step, because all the steps is already ran before, so we can see the output anyway. So I mean, I wouldn't really hesitate. So next part is the basic QC. So what is QC? This is the quality control. So this is very crucial steps in single cell RNA-seq analysis, and also the single cell techniques. So QC is basically involved in assessing the quality of the data at various stage of experiment, ensure the results are accurate and biologically meaningful, right? So which QCs you can do? First is you can do library preparation QC. So this is not really computational stuff. This is for your experiments. So what you can do is you can check the cell viability, assess the viability of the cell before the library preparation, because you're going to make a single cell library. So if your cells are already dead, then or the cells are unhealthy, then this cell is not going to be, converted into the quantification with the good quality, because all the gene expression distorted in some way, or the genes are removed, or genes are all, excluded in a certain external exposures, right? So you have to check the cell viability first time. And after that, you have to think about the library complexity. So you have to evaluate the complexity of libraries to ensure they're representative of transcriptome, right? What this means, if you have very low complexity of your transcript in your library, then you're not going to get any information out of your information. So out of your sequencing, right? So low complexity can, indicate some issues with the library preparation. So this is another things you can do quality check in your library preparation stage. And once you've done that, then you're going through the sequencing. So sequencing stage, also you can do QC. So first thing, you can check the sequencing depth. This is basically the procedures where adequate numbers of reads have been generated to capture the expression of the genes in the cell. So sometimes low sequencing depth can result in incomplete data. This is, of course, right? And the read quality, this means that if you look at your sequence read, sometimes quality are very poor. So you have to go through the QC using the faster QC files to detect any issues with base quality, or any adaptive contamination, or any anomalies coming into your library. So once you've done all of things, then you are finally getting into the a single cell data set. Then you're going through the sort of the cell level QC, which is the first one is the gene detection rate. You're basically examining the percentage of genes detected in each cell, and you're going through the low genes detection rates, may indicating poor cell quality or inefficient library preparation. And the second things you can check is the mitochondrial gene content. So high level of mitochondrial gene expression in a cell can be a sign of poor cell health or contamination. So you basically monitor mitochondrial gene, then you can identify the low quality cells. And after that, you can basically detect the multiplets or doublets. So why these things really matter? Because you're supposed to get the single cell out of your experiment, right? So when you make a single cell data set, we basically get some, the platforms you captured one cell in a one droplet, right? But sometimes within one droplet, you have the multiple cells within there, then that droplet cannot give you the good quality of the data set. So you have to remove all the situations where two or more cells within a one droplet captures, then these things we think is a sort of low quality or artifact in your data. So in that case, we have to remove those cells before you processing the data set. So we call them multiplets or doublets, right? So multiplets is many different cells they capture together within the droplet. And the doublet is the two cells mistakenly identified as one. So this is another problem. Okay, so once you get all that information, then we go through the actual QC. So let's look at, before we going there, it's almost there. I can see the RAM is dropping because it takes the, where is it, quantification, then it checks some bus tools information. So it's nearly there. Okay, so once you've done that, then we can load the Python libraries to process the data. So let's say like you deal with the numbers, you have to import the library called NumPy, and you also load the pandas, which is the data frame library, and you load the data. It's the Scampi, which is dealing with all the single cell stuffs, right? So when you import the library, you say import the NumPy, but as MP. So you can use the abbreviation to make much shorter in your command line, right? So MP or pandas is PD, Scampi is SC. You can also import the N data. You can also import the scikit-learn, and specifically you import the modules decomposition, right? From there, you import the SVD truncated version. Okay, also you can import other, plot-related things like a matplotlib. In a Python programming, you can use the verbosity, refers to level of detail, clarity in code, especially how explicitly concise the code is written, right? And verbose code is one that contains more words, statements, annotation to make code's intent, functionality more explicit, where the less verbose code is more concise, often using few words of statements, right? So we can basically set up the verbosity when you run the code, right? So here we set up the verbosity 3, which is I want to get all the information even with the hint, right? But if you want to run your code with very concise and quietly, then you can say less information, right? So like a 0 or 1, right? So you only get error or warning level information. But if you set 3, you can get error, you can get warning, you can get information, you can get hint. All of the complete package. Okay, so before we move on to actual QC, you have to learn about the N data. N data is standing for the annotated data. It is a data structure used in single-cell RNA sequencing, particularly in the context of Scampi library in Python. N data is designed to store, manage single-cell data efficiently while allowing for the integration of various types of information and metadata. It is versatile, organized data structure that simplifies the handling of single-cell RNA-seq dataset. And in the N data, the data structure used in the analysis with the Scampi. So it specifically used the N data OBS, which is the observation. And the other one is the ndata.var, which is the variable, not for the football. Sorry. Okay, so ndata OBS, observation. This is annotation of observations. You can store many different layers of information like a cell identifiers, cell type labels, sample information, quality control matrix, clustering assignment, experiment annotation. So all of the information not automatically stored in this N data observation. You have to add those information if you need it, right? Or if you process it, then you can store that information into this system, into this structure, sorry, not the system. Okay, so this is basically the pandas data frame, and this is allowing for each manipulation, querying and visualization of cell level metadata, okay? And data VAR, which is the annotations of variable. This is the dictionary-like components of N data object. So this is the storing annotation related to genes, right? So as you can see here, so this is the cell level information, cell level metadata, and this is the gene level metadata, right? So observation and variables, right? So it has the sort of the distinction between these two. Okay, so as I mentioned, that this is the gene level metadata, so it has the gene level information here. like a gene names, gene annotations, gene biotypes, or custom annotations, all things you can describe for the genes. So how can you use this information? We use for the cell type annotation, we use for the quality control, we use for the visualization, and we use for the gene annotation and filtering. All of that information you can centralize by this N data. And if you have further information, you can go to this link. You see the concept how N data is working. Okay, so next is we're going to use the input data and learn about which input data we use for our tutorial. So input data we're going to use is the 3K PBMC from the healthy donor. And this is a free data we get from the 10x Genomics. So what is PBMC? This is peripheral blood mononuclear cells, shortly PBMC from the healthy donor. And this is the primary cells. What is the primary cells? So we have the primary cell, we have the secondary cells. Secondary cells is normally you get the data, you get the cells from the culture, right? So you get a cell, you culture cells, many different passages. So that case we say secondary cells, but primary cells you get the cell from the original donor, right? Original humans or original animals or plants, you get a cells from the source, right? And primary cells, and it has the relatively small amount of RNA, so 1 picogram RNA per cell, which is very tiny, right? So within these blood cells, we have the 2,700 cells detected and sequenced by Illumina platform, and we get this amount of reads per cell, right? And we have about 100 base pair read one and 8 base pair barcode, index barcode, and 14 base pair index gem code barcode, and 10 base pair read 2. And the analysis run with we set up the 3,000 because it's around 3,000, so we set up that. Okay, so the first thing you have to do is you import the unfiltered matrix. So we say import or we say load, right? So result file in an H5 format, as I mentioned before. And we load this data called the N data library, then dot read underscore H5AD. So what does this mean? We use this function, which means that the reading H5 file, like here, right? And why is dot? Because you retrieve this function from N data, right? So you can hover your mouse, you can see the information, right? So what this means, you can read the H5 format file into your system, right? So once you read this file is stored as a data object, right? In an A data object, you have the var, the object. Then within this var, you can store the gene ID from the index of the values, right? Then once you've done that, then you read the table file using the pandas read.csv, right? So this is two different things. So N data is the library for single cell data set. You read the H5 file, but PD is the pandas file, which is reading the text file. So T2G txt. Then you get a T2G file. This is again, you have all the gene ID into the index because you want to merge this information between these two, right? So you can basically map this T2G gene name into a data var gene name, right? So you make sure all the things within together, right? So a data var index and a data var gene name, right? And after then you have var name make unique because some of the gene names they have very duplicated entries. So you make sure all the things within very unique entry. So once you get the A data, then you type the A data, then you see how many information you get from loading this information. So how many cells you get? 250,000, and you have how many genes and number of R's? 60,000 genes, right? This is way larger than your expectation, because we say we get around 3,000 cells, but here it's more than 8 times greater than expectation, right? Because this, I mean, as I mentioned, that we loaded unfiltered matrix, so this is the numbers you get from the droplet rather than actual cell captures, right? So if you go through the filtering, then you remove many of droplet, which means the droplet containing the cells, you get it into the real numbers, right? Okay, within the A data, if you type adata.x, then you retrieve this X object, which is this parse matrix, which is like an Excel type matrix, but it's partially store the information within this matrix format. And A data OBS, it contains the barcodes. This barcode is basically meaning each cell, right? So each cell has these barcodes, so we can distinguish between the cells. So it has around 250,000, right? Because it's the total number of droplets we can get from the 10x. And A data var, this is the gene matrix. So as you can see here, 60,000 genes. See here, 60,000 gene name and gene ID. So we looked at all the data structure related to single cell data set and also the pseudo alignment process. So this is sort of the very first part of your single cell analysis. And we finalized the first part. Then now we continue to discuss the downstream analysis. You're going to go through the single cell analysis. So see you on the next video.